And welcome to this year's Karlovitz Lecture, which is co-sponsored by the Georgia Tech Honors Program and the Georgia Tech College of Sciences. I'm Greg Nobles, the director of the Honors Program, and I'm always very pleased to see all sorts of people come to these events, students and faculty, members of the staff, and perhaps, for all I know, people from the larger Atlanta community, and that's just great. It's good to see people come together for events like this. So thank you all for coming on what is, I must realize, a very pleasant day outside. We'll have a very pleasant hour or so inside, I'm sure. We'll be bringing nature inside. I have to say that events like this don't just happen. And this one wouldn't have happened without the very hard work and the energy and organizational skills and, frankly, just force of will of my colleagues, Monica Hawka, Nicole Leonard, and Nairuz El-Amali, all of whom really did the heavy lifting, the hard work to put this together. So I especially want to thank them. And now, having thanked everybody, I will invite everybody to take your electronic devices and silence them, or indeed turn them in the off position. You can actually go text and tweet free for 1 hour. It'll do us all good. I always like to say a word about the man for whom this lectureship is named, Les Karlovitz. Les was the dean of the College of Sciences and Liberal Studies, COSAL, as it was called, back in the 1980s when I came to Georgia Tech. He was a mathematician, but a man of very broad vision and very broad interest. He had the ability, I think, to intimidate assistant professors. I happened to be one of them at the time. But he also, I think, had the great ability to bring people together, to welcome people. He set very high standards for his faculty. But he also created opportunities for his faculty to come together. And so we would have historians talking to chemists. We would have biologists talking to literary scholars. And that's the kind of environment that Les Karlovitz tried to create while he was at Georgia Tech as Dean of the College of Sciences and Liberal Studies. Sadly, Les Karlovitz left Georgia Tech and then died in 1990. And so this lectureship has been named in his memory and, frankly, yes, in his honor. And I think it's a great opportunity for all of us, again, to come together at least once a year and to talk about science, but to talk about science not just as scientists. And I think one of the things that Les Karlovitz helped make possible here was what I like to call accessible science, where people in scientific disciplines get to talk to those of us, frankly, who are not in those disciplines and explain what's going on. And so I think that's a great opportunity we have this afternoon with this lectureship. Les Karlovitz was the former dean of the College of Sciences and Liberal Studies. That college has now transformed, or been transformed, frankly, and now we have a College of Sciences. We have a wonderful dean of our College of Sciences, Paul Houston, and I'm going to invite Paul to introduce our speaker this afternoon. So, Paul.
Thanks very much, Greg. It's a real pleasure this afternoon to introduce our Karlovitz speaker for 2011, and that is, as you've heard, Dr. Erich Jarvis, who is an associate professor of neurobiology at Duke University. He's had an interesting history that I think may be inspirational to some of you. Many of you may have read about him, but let me just try to recap some of the features of it. He grew up in Harlem, New York, and was the son of two musicians. Early in his life, I guess in grade school, he wanted to be a magician. That was his first love. But as time went on, he decided that there were other things in life as well. And so he went to the prestigious High School of the Performing Arts in New York, and there he fell in love with dance. So time went on, and he became a better and better dancer. Did a little science on the side, I presume. But by the end of high school, he was thinking, gee, a dance career wouldn't be such a bad thing. But, you know, I really ought to have a plan B. So maybe I'll apply to a few colleges just to see what happens in case my dance career doesn't pan out. So I applied to a few colleges and got in, and as graduation day came along, a couple of days before graduation, he actually got a call from the Alvin Ailey Company to come and audition to be a member of that company. And so that was a time for him that was a pretty difficult decision. He had to decide whether to be a dancer or to go on to college and do other things. And after some soul searching, he decided to go to college. Later he said about it, I realized that I would have a bigger impact on the world if I became a scientist. So he went off to Hunter College, and there his inspirations were two people I like to see live together, Barbara McClintock and Albert Einstein. So that led him to McClintock on the one hand, led him to biology, and Einstein, I guess, to mathematics, because he double majored in those topics at Hunter College, but he minored in chemistry, and that's a good thing from my personal point of view. So he did well at Hunter, and then went on to get his PhD at Rockefeller University, where he stayed on as a postdoc in the general area of neurobiology. And after some years there, he then joined Duke University, where he is now as an associate professor. Along the way, he won almost every award that a young scientist could hope to win. He won the NIH Minority Biomedical Research Traineeship, and then won first place in at a conference for the paper he presented there. He had a Ford Foundation Predoctoral Fellowship. He won the NSF Alan T. Waterman Award. I don't know if you're familiar with that award, but it's given, one a year is given to the most promising scientist under 35, so it's a great honor to get that. So that was from NSF, and then the NIH Director's Pioneer Award. Now with NIH and NSF behind you, you've got a great career, but that wasn't enough, because as you can see, he was awarded just a few years later a Howard Hughes Medical Institute investigator position, which he still holds. But probably the most important award to some of you will be that he was already featured in a Nova Science Now documentary. And if you want, you can go to the web and find this documentary and see the story of Erich's life, including some clips of his dancing, which are very impressive indeed. So it gives me great pleasure to welcome Erich to Georgia Tech, and we look forward to his talk on brain evolution, how birds and humans learn to sing and talk.
Well, thank you. Thank you.
And so pay attention to these two themes throughout the talk. There we go. Is that showing up? No. And I encourage you after the end, students should always have questions, and you're allowed to have questions about either one of these themes, even though 90% is about science. And I was asked to put the slides here, but you really summarized it all anyway. But going all the way from my early years, yes, as a magician, when I was a young child, to a dancer in high school performing arts, undergraduate student, where there I was quite well by some programs that helped fund research in the laboratory for undergraduate students, and stayed in the lab for 4 years, the same lab as an undergraduate student, and managed to get 6 scientific publications out of that, 2 which I was first author. And that was in bacterial molecular genetics. And then from there, I had another epiphany. You know, the first epiphany was, do I want to be a dancer or a scientist? My plan B really wasn't so much about not thinking I could make it as a dancer, although evidently hadn't come knocking at my door yet. So I really didn't know. The plan B was really, like you said, I thought I could have a major impact on society. So then here comes another epiphany. It's going from that to graduate school, and I was trying to decide, do I want to understand how the brain works or the origins of the universe to be an astronaut? Those are the 3 I was choosing between. So I chose the brain because I felt it was more down to earth for me and the type of training that I received. And went on to graduate school at Rockefeller University in New York, where I entered the laboratory of Fernando Nottebohm studying vocal communication in songbirds. And why did I choose songbirds? not because it was a link to, artistic singing abilities. My singing abilities wasn't that great. My dancing was better, right? But nevertheless, it was that these animals seem to have the ability, that is some songbirds and parrots, to present complex behaviors such as vocal imitation that we don't find in non-human primates or in other common laboratory animals. And that's why I did that. But you'll see halfway through this talk that I did come back to my dancing life in my science. Then I went on to Duke University after my postdoc, and I've been there for the last 12 years, but I have a lab, a decent-sized lab, and I've graduated a number of graduate students myself now and postdoctoral fellows who have their own lab, and given a number of lectures as well on this topic. And I always love to give a lecture to an audience like this where students come from a diverse background, because it's a challenge for me to communicate a complex topic about how the brain works. If you're going to choose between the universe and the brain, that's how complicated it is, but yet to a general audience. And yes, I still dance. I do African dance now, and I just joined the salsa dance team. So I always encourage students to have something else in their life than their main career, because it helps you balance out your life. All right, the topic here, vocal learning, or vocal imitation, or whatever you want to call it. I like to call it production vocal learning. It's a very rare trait. Only 5 groups of mammals are known to have this, and 3 groups of birds, and they're listed up here. And it's recently been shown in elephants. Can actually imitate truck sounds along the road. This was shown in some elephants from the zoo. And some African elephants imitating Asian elephant calls that they brought together in the circus. There are 3 groups of birds that have this ability. The word parrot has become synonymous with vocal imitation or mimicry. Many people don't realize that hummingbirds have this ability as well, although they're mistaken to be producing insect-like sounds, but they have these high-pitched vocalizations, which I'll play for you a little later. And then songbirds, like your mockingbirds and your canaries, your pet songbirds. Now, vocal learning is different from other types of learning that we humans sometimes confuse because of our daily speech experience. That is auditory learning. Auditory learning is the ability to make sound associations with the environment. And I'm going to give some examples. Dogs, for instance, can understand the word sit. Right? They can also understand siéntese or salado. Right? Even though many of you couldn't understand, or some of those words. Right? Because a dog and many other animals can even learn through auditory learning in their brain human speech sounds. And not just simple one words. They can learn, come here, boy, fetch the newspaper, get this toy or that toy over there. All that occurs through auditory learning. And most vertebrates have that ability. Not necessarily learning sounds of other species, but still, they learn. Whereas vocal learners can actually learn how to say sit, like your own child or your brother or sister. And so we have been asking what is different about these species, when I'm talking about these comprehension auditory learning, that have the vocal learning ability and everybody else, and everybody that has vocal the auditory learning ability. Now I mentioned auditory learning, but there are 2 kinds. The kind that I just explained is called comprehension auditory learning. There's something called usage auditory learning, and that is all species have the ability to produce vocalizations. Most species do. Some reptiles don't, and some fish don't. But most vertebrates do. What they learn to do by usage learning is a dog, well not a dog, a chicken or a vervet monkey will produce a particular alarm call that is genetically innate. The actual sound is genetically innate. But through auditory and visual experience, it will see the adult produce this particular alarm call not to a rock on the ground, but to a snake or to an eagle in the sky. And so it uses that, it learns how to use that innate sound in different social contexts. But that is still an example of auditory learning. All right, so I said it's rare, and here's a family tree two different views, one of mammals, the other of birds, of showing vocal learners highlighted in red here. And so what's interesting about all vocal learning animal orders to date is that amongst primates shown here, only humans have this trait. Chimpanzees, macaques, those are monkeys and so forth, squirrel monkeys, don't have this ability to imitate sounds. They do have auditory learning, but not vocal learning. Bats, and I mentioned elephants, dolphins, are cetaceans. So it's been argued that amongst mammals, this trait has evolved independently of a common ancestor, because lions, tigers, and bears don't have it. Why would these species that are far distantly related to us have it, and we don't have a common ancestor with that ability? That is a close relative, like I said. Then birds, we have three groups here: the hummingbirds, parrots, and songbirds. And you can see that in this phylogenetic tree that they are also separated by other species, not bolded here, that are the vocal non-learners. What's interesting here is that there is a close relationship between parrots and songbirds in a more recently derived phylogeny here. And there's this group in between them called the sub-Oscine songbirds that don't have this ability. And the sub-Oscine songbirds are just as closely related to what we call true songbirds, the vocal learners, as chimpanzees are to humans. And one argument has been is that if parrots are really that closely related to songbirds, then maybe there was a common ancestor that had the ability to imitate sounds, and sub-Oscines lost the trait, where songbirds and parrots retained it. It's like saying there's a common ancestor of humans and chimps that had the trait. Chimpanzees lost the ability to produce spoken language, and humans still have it. So imagine that. What's interesting, whether or not this is a loss versus an independently gained trait, it certainly shows that there's convergence in all these vocal learners that have been studied, that we all depend on auditory feedback to maintain our vocalizations. Without the hearing, our vocalizations will deteriorate. It's called the speech sound deterioration disorder. We have critical periods, songbirds, parrots, and humans do. We learn best early in life to imitate sounds. After puberty in humans and in birds, you can't learn to imitate as well. And you get dialects as well in different locations. We get different languages. And possibly autism. Birds are now being used as models for autism, as vocal learning animals. Now, looking at the birds itself, there isn't really a one-to-one correspondence with the actual song repertoires of these birds in human language, but you can find some types of parallels that are closely related. And this is how I broke them down here. For humans, we talk about speech or singing. They're really using some of the same brain pathways. They're not that different for spoken language. So I really like to think of what I'm talking about today as spoken language, as opposed to perceptual language. Like, come here, boy, fetch the newspaper. That's not what I'm talking about. I'm talking about the ability to say, come here, boy, fetch the newspaper. All right, as to what's unique. Songbirds, we just call it song. And then there's a bout of song that we usually, I would say it's akin to a whole sentence for our speech. And those bouts can be broken down into stereotype motifs in songbirds, or words in human speech, and then individual notes or syllables versus phonemes, the actual ba, ta in human speech. Now, most vocal learners do not have as complex a repertoire of vocalizations as humans do, and aren't as prolific as we are. We are the most prolific vocal learners. But there are some vocal learners who can start to come closer than others, particularly the African gray parrots. And I'm showing you an example here, but before I play this video, I want to say that most vocal learners, whether they be a dolphin or a whale or a canary, use their learned vocalizations to attract mates and defend territories. The more variable the sound that they learn, the more competitive they are. Right? But what has happened to some vocal learners like us and the African gray parrots is that we use those learned sounds for more semantic communication. And so here in this example, I just want you to listen to this video. Turn this up. so multiple things going on here, because when I like to present this for a number of reasons, and you'll see why. One is that this animal is reciprocally communicating with a human. Okay? It's understanding the question like a dog would, but now it's actually talking back. All right? Second, what it's talking back, it has learned from another species, that is a human. In this case, the word one. All right? Now, Alex was a famous parrot. He died several years ago, and Irene Pepperberg, who's the scientist who raised him and studied him, had him counting up to 7 before he died. But why do I show you this example of 1? Because people think, oh, 1 is simple. Well, the way Alex actually counts 1 is that he adds 1p plus 0 under the cup together to get 1. And before this study, it was argued that non-human animals don't have the concept of 0. Well, Alex had the concept of 0. And this is what you can learn from an animal who can imitate human speech. If you had 2 p's and 3 under the cup, you would actually count and add up to 5. So it is possible for the brains of these small animals to actually have some kind of complex communication. So with that introduction, what I decided to present to you today about this topic is 3 different areas of research in my lab. One is actually trying to identify the vocal learning brain system in birds and comparing with the human literature and some work we've done in our own lab also on humans. And the other is from what we've learned from this comparative analysis here, come up with a new theory in the past 2 years, what I call the motor theory of vocal learning origin. And then I'll end off with some of the genes that I think are involved in setting up these brain pathways. So starting out actually with some genes, it was the genes that led us into identifying more of these brain pathways than what had been done in the past. The gene that we studied most is called ZANK, or another name for it is called ZIP268, EGR1, nerve growth factor induced G1A, that's what these acronyms stand for, CRUX24. At the time we cloned it from the bird brain, a number of laboratories had also cloned it from humans, mice, and other species, and those laboratories gave the gene a different name each. So we decided to be fair to those 4 labs and just called it Z and K, ZANK. And it was appropriate because the protein shown at the bottom here is a transcription factor that regulates other genes and has what we call zinc finger binding domains, so you see these here, that sit in the promoter region, these are regulatory regions of other genes, and will either turn on the synthesis of their mRNA or shut it down, using those zinc finger molecules to sit in the DNA. So for those who haven't heard transcription factors before, they're basically proteins that regulate the synthesis of other genes in the brain, or in the body in general. What was interesting about ZANK at the time it was discovered is that in the brain, this gene is responsive to neural activity. When neurons fire a lot of action potentials, ionic currents across the membrane, it induces a signal of cascades in the cells that induce the mRNA for this protein. So we had started studying in songbirds, and what I found, oh, someone's supposed to turn it off? Is that when birds sing, and I'm going to show you here's a canary song. This act of producing that learned song causes a huge increase, a robust expression of the mRNA of this ZANK gene in the canary brain. Shown here is the brain section in what we call the sagittal view. This is the cerebellum behind in the back of the head, and this egg-shaped region up here is the telencephalon, or the forebrain. And this is from an animal who was singing. It's like an MRI image for 30 minutes, what we call a behavioral molecular mapping image. And here is an animal who was quiet. And you can see that there are these structures in the forebrain that we call song nuclei that light up with the singing behavior. And these structures we know are responsible for producing the learned songs, one called HBC here, and the other, the area exon striatum, for learning the songs. Before we discovered this phenomenon, a lot of people were trying to look for gene expression of this particular gene in these song nuclei, but playing song to the birds or doing some kind of classical conditioning, Pavlovian training paradigm, pairing song with shock, and couldn't get the gene to be expressed in the brain in these song nuclei. But what we found is that we just have to let the bird sing. And that simply just causes the induction of the gene. And it's a lesson for the students here, and that is sometimes just let the animals behave naturally as they want, and you can learn more from them than if you try to do all this crazy stuff. So we found that that was one song bat. We found that the more they sing, shown on the x-axis here, the higher levels of gene expression we see in their song nuclei. And so there's a one-fold increase for every song belt that the bird sings in this structure called Erich, for example. And this diagram, this image shows you those same two brain sections here, but now showing you what happens when you play song back to an animal that is hearing a song, like hearing, come here boy, fetch the newspaper. You see brain areas showing activation relative to the silent animal that we now know as the part of the auditory forebrain. I'm showing you my arrow here. But these actual song learning nuclei don't show any gene activation. That is, they don't seem to really be highly active in the processing of the sound. And we now know that is the case that when you deafen them, that is, they can't hear, you see this hearing-induced region is now negative. It doesn't show any increased expression. But these song nuclei actually still show expression of the gene. It's less than a hearing intact animal. That's because the deafened animals tend to see less. So this showed that there was a distinct separation between areas of the brain that control the actual singing behavior and those that actually process the actual sounds. So why is this interesting? Well, we know that species that can't imitate sounds do not have any of those song nuclei in the forebrain. And we knew that for songbirds, but we didn't know if that was the case for parrots and hummingbirds. And we knew that was true for humans. And so the question became, are songbirds just something strange, or do the other vocal learners have such brain areas? So we applied our similar technique on hummingbirds and parrots as well, parakeets, which are basically small parrots. And just in case you think that the brains of these small animals can't do anything complex, this hummingbird is about that big. It's about 3 inches long, okay? And it has a brain that's at smaller, about the size of my tip of my finger there, all right? And this is what it produces. I'm trying to follow the song here. This is the sonogram. Okay. So what you're seeing here is a sonogram of the sound that the bird just produced. Along the x-axis is time. There's 20 seconds from the beginning to the end. And the y-axis here is the frequency of the sound. And each of these individual syllables here are basically notes in the song. And you can see, just by looking at the structure, they're quite complex and varied, and they morph from one form into the other. All this is learned. You can't get a dog or a horse or other species to actually learn how to do something like this. So what about the hummingbird brain? What about a parrot brain that didn't take human speech? We applied the same approach, and what we found is that, like in songbirds, when parrots, shown in the middle here, or hummingbirds, shown at the bottom here, hear species-specific sounds, you get auditory forebrain areas showing hearing-induced gene expression. When they vocalize their songs, or imitated sounds, you get these other forebrain areas now lighting up, such as in the anterior forebrain here in the parrot, as well as in the hummingbird. And some of these structures, like here, this VLN nucleus in hummingbirds, looks quite similar to this HPC nucleus in songbirds. Somewhat in a similar location, just pushed more to the side of the brain. That's why there's smaller tissue here. And when you put these diagrams together in three-dimensional images, what you come up with is as follows. Is that on the top we have these several different views of our phylogeny. I'll get back to that later. But we have a 3D reconstruction of a songbird, parrot, hummingbird, and a non-vocal learner, such as a pigeon or a chicken, or suboscine songbirds. And we find that in all species of birds, vocal learners and non-learners, they have this auditory forebrain pathway, I color-coded in blue here, that is responsible for processing complex sounds. right? So that is not unique to vocal learners. But they all have, the vocal learners that is, have exactly 7 brain nuclei in their forebrains that I color-coded in red and yellow here. 3 of them sitting in the front of the brain, the red ones, involved in the imitation of the sound, and the 4 other in yellow, sitting more caudal in the brain, that is closer to the back of the brain, that's involved in the production of the sound and sends out these connections from the forebrain to the motor neurons that control the vocalizations. What's interesting about this is that whether you look at this phylogeny here from the 1990s or this recently recently derived phylogeny based upon DNA evidence, is that you have to argue that there were at least 2 independent gains of 7 brain areas in the last 65 million years for this trait. All right? Imagine that. It's like evolving wings multiple times in birds, bats, ancient flying dinosaurs. Now evolving a brain pathway that can imitate sound, including human speech. Multiple times, exactly the 7 brain nuclei, similar connectivity, not identical. So when we published this at the time, I got a lot of communications from religious groups. So I argued this supports intelligent design, or the existence of God, because how could this happen multiple times like this? And I didn't have an answer. My answer was just as good or bad as that. I didn't have an answer for a number of years, but before going on to an answer, I wanted to take an objective look here and ask, comparing to humans. Here are my 3 hypotheses I typed up here. Is that there are either multiple independent gains of these 7 brain nuclei with similar connectivity, or multiple independent losses. Maybe there was a vocal learner 30 million years ago, and owls and swifts and the eagles and the pigeons and the suboscine songbirds, they all lost it. There was mass extinction of the trait. Okay? Okay, that's okay. Or you can think of that for humans and dolphins as well. A lot of species just lost it. And maybe we haven't tested all of them as well. Or there was a common ancestor on this tree here at least, and maybe just independently derived twice. What about humans? So we had to get rid of some dogma in order to compare human brains with songbird brains. And this slide shows us a few lessons. So to your left is the zebrafinch brain, the common laboratory animal we study for vocal learners. And it shows the forebrain here, where we have those song learning nuclei, the cerebellum, midbrain, spinal cord. Here is a human brain. The first lesson we learned here is that here is the zebrafinch brain to scale with a human brain at the right corner. Brain size doesn't matter. You can fit roughly 3,000 zebrafinch brains into one human brain, or a big person at least. The second lesson learned here is that the cortical regions in human are folded, whereas that's not the case in a zebrafinch. So folding, the noodle-like shape as we like to think of it as children, doesn't matter either, even though that adds a lot of extra surface area to the brain. Brain size is more important for animal size. Imagine fitting a big human brain into an animal that size. Not possible, right? What's really making the difference is the presence or absence of a neural network. And to show you what that neural network is, I have to get rid of some dogma that's still in your textbooks today. All right? And that is shown in this diagram here. The current, or let's say the view of vertebrate brain organization that has been around for the last 100 years was developed by a guy named Ludwig Edinger in Germany at the turn of the last century, from the late 1800s, early 1900s, where he and his students were trying to do like Einstein was doing for physics, was coming up with a universal theory of everything, but in this case, the universal theory of the brain. And their argument was, taking Darwin's recent view of evolution, that evolution occurred not like Darwin thought, but in a progressive fashion, scale and natural. And that the evolution had a purpose to it. And that purpose was to generate man, particularly men, not women. And if you look back on those old texts, talking about culture and science, European men, of all things. So you see in these early comparative neurobiology papers, these trees with, Europeans at the top and so forth, and the fish at the bottom, and frogs and so forth. And so, and interesting, but the science wasn't as objective as we would like, and the argument was that in the human brain, you can trace back an old structure that they called archostriatum, we could now call the amygdala, involved in emotional behavior, and that was present in fish. And then fish gave rise to a new structure called the paleostriatum here in amphibians, that is. And this is now we call the globus pallidus in the human brain. When that gave rise to a new structure called the neostriatum, and we still call it the neostriatum in our medical textbooks. It's called neustriatum because reptiles invented it. And they passed that on to birds, which is this region here. And then birds invented something new called the hyperstriatum, for overgrown striatum. All these purple labeled regions were called striatum, meaning basal ganglia structures, thought to be involved in primitive behavior. At that time, these comparative neurobiologists knew that the parrots could imitate, but it was sort of strange for them. Maybe parrots somehow evolved some type of imitative structure in their basal ganglia. So I won't get into all, I don't have time, this is a whole nother lecture to get into the details of what made us say that this view is totally wrong, but it is. All right? And that is, from my lab and many other laboratories, my lab just recently, but other laboratories starting from the early 70s and through the 90s, have looked at comparative gene expression profiles, neural connectivity, brain lesions in birds compared to humans and other mammals, and have said that these terms like neostriatum or neocortex, meaning new cortex only in mammals, is totally false, because it was thought that birds hardly had any neocortex. And the view now that we know is that birds, like mammals, have a large cortical territory that makes up most of the forebrain. It's just organized in large cell clusters, like this one here, as opposed to layers. And if the connectivity is similar, you can imagine taking two layers like this in the human cortex, and they have connections between the two, well, 6 of them all together, and now ball them up and put all the cells on from one layer over here and all the cells in one layer over here, but keep the connections similar. Okay? That's how you should think of the bird brain and the human brain. All right? And out of those two different structural organizations, layered and clustered, you can get vocal imitation. And how does that work? All right, so now we're going to look through a section of the songbird brain and a section of the human brain and compare these structures. So in green, I have the cortical region labeled, and I have the basal ganglia regions labeled in purple and in blue. By the way, the basal ganglia is not only involved in primitive behaviors, it's actually a complex region involved in complex cognitive behaviors. And I asked the question, is there anything in the human brain that when damaged through stroke, trauma, and so forth, leads to similar symptoms of what happens in the songbird brain? And the answer is yes. There are parts of the anterior cortex, the anterior striatum and thalamus, that when damaged leads to what we call speech aphasias. We can still speak, but we can't learn new song. I mean, well, in birds, if those areas are damaged, they can't learn new song. In humans, we can't learn how to sing new songs or speak. So this is the famous Broca's area in the anterior part of the forebrain, all the way up to what we call dorsolateral cortex right here. And in non-human primates, non-speech areas to which these areas are adjacent to, which we can determine connectivity, are connected in what we call cortical basal ganglia thalamic loops. This loop shown here. Such a loop is similar to this loop I have in these white arrows here in the songbird brain. All right? So I'm arguing that this is an analogous pathway. I'm not arguing homology here, but somehow independently, maybe independently derived. In non-human primates, these motor pathways project to the cortex that I just showed here in the human brain. And I'm mapping onto the human brain. Like this pathway here projects to the motor pathway in yellow here that produces the learned song. But in humans, I don't think it's just Broca's area here that causes what we call speech aphasias. This is now what we call a frontal section through the human brain. You're looking at the cortex here, gray matter, and these regions down here at the basal ganglia. There's a whole strip of cortex, I don't have time to get into detail now, on the left and the right side of the brain that when damaged leads to either verbal aphasias on the left side, that is, we can't speak well, but we can still sing, or verbal amusias on the right side. That is, we can't sing well, but we can speak. All right? That is, there's a dominance in the human brain for this whole cortical strip here. And such dominance also exists in songbirds. So the canary is left-sided dominant like a human for its vocalizations, whereas a zebrafinch is right-sided dominant. So I'm calling this the language strip. And there's a region of that's analogous to those red-labeled regions I talked about in the song-learning birds. What about those yellow-labeled regions that control the production of what I'm speaking, the production of learned sounds? Well, in the human brain, there's a region called the face motor cortex, shown here. And it's the only region in the human brain that we know the connectivity for a vocal region. Because in post-mortem patients back in the 1950s, a professor named Kuipers had looked at degenerating fibers from this region and found that it projects to the region called nucleus ambiguous in the brainstem that controls the production of innate sounds down there, but now it's controlling it from the cortex to control learning of those sounds. So when face motor cortex is lesioned, what happens is that we lose the ability to speak at all. We can cry, we can scream, we can go ah, innate sounds, but we can't actually produce learned speech. This is analogous to the lesions in those yellow-labeled regions in songbirds. When damage causes a problem, you can't sing at all. And in non-human primates, if these areas become damaged, that is the face motor cortex, they can still produce their innate calls. Likewise, in a chicken or a pigeon, when an analogous area becomes damaged, they can still produce their innate calls. So this region of the brain and its connection is what I consider one of the most critical traits that led to the evolution of vocal learning or speech in humans and song-learning birds. And it's a connection that we share with parrots and songbirds that is not shared with chimpanzees or chickens. So there's an auditory pathway that can be found in all vertebrate species that goes from the sensory ganglion, from the ear carousels to the brainstem to the midbrain to the thalamus, all the way up to the cerebrum, that I argue is involved in auditory learning and is necessary for auditory learning. So I'm going to summarize this part here to say that we humans have also this auditory pathway, includes the famous Wernicke's area involved in speech perception, that I argue is actually homologous to an ancient auditory pathway found in all, at least land vertebrates, color-coded in blue here. And that is what we share with the birds. And this is why Alex can understand, Alex, how many? What color? Or a dog can understand, come here, boy, fetch the newspaper, in terms of human speech. And that humans have come up with the face motor cortex region and a strip of cortex in the basal ganglia, the thalamic region, involved in imitation for the red regions and the production of those imitated sounds. Independently, maybe independent, I think independently of the song-learning birds. So how did that happen? And my second topic here, I hope I'm doing okay with timing, but we'll get through these. All right, is the example. I don't think it's intelligent design, but I didn't have an answer to even how humans and songbirds, 300 million years apart, could come up with a similar solution. And that answer came in an accidental discovery in another experiment we were doing, trying to look for birds' brain areas controlling migratory behavior. And in this case, this is a bird either hopping or flying, trying to migrate. And we found that when the birds are moving their limbs, what we get, instead of gene activation with singing, like in these song 7, song 6 of the song nuclei here, with limb movements, we get gene activation in 7 brain areas surrounding all 7 song nuclei, or adjacent to them. An example here is shown, this is the entire striatum region right here of the brain, an area homologous to the striatum in humans. Singing activates the area X nucleus, whereas hopping activates one cell layer removed around that nucleus here. And we found that is the case not only for songbirds, but also for parrots, that the regions controlling their limb movements are surrounding areas or adjacent to regions that control this song or the learning of the prediction of the innate sounds. And here is your hearing activated regions. So, and we found the same thing for hummingbirds, which I'm not showing here, but we went to a non-vocal learning species, got them to do hopping, in this case a dove, which is basically like a pigeon, and we could see areas of the brain in the same location activated here. There are actually 1, 2, 3, 4, 5 of these regions right here, sitting on top of each other, but no holes of absence of expression of the song nuclei, confirming that the pigeon or the dove here doesn't have the song learning nuclei. We found 7 such regions in them. Well, what about the connectivity of those areas surrounding the song nuclei? We found that their connections are quite similar to the song nuclei themselves, forming a loop here, projections out of the forebrains to other motor neurons that control limb movements. There's some differences, like shown in this red arrow, but for the most part, the connectivity is similar. And this led me to ask, is there motor learning? Are these brain pathways controlling motor learning? And I come up with a theory that that is the case, that animals do have complex motor learning skills, including learning how to dance. Most animals do. And this is shown in a famous cockatoo called Snowball. I'm going to show you a video of him. Oh, let's see. Oh, here we go. Just bear with me. Some video I have to get offline. Has some introduction here. So the difference, what you'll see about vocal learning species who can learn how to do movements, is that they can synchronize music better than non-vocal learning species.
This is so much better than Dancing with the Stars. Tell us very briefly what Snowball can do, and then we'll witness it for ourselves.
Well, Snowball can dance to a beat, and he actually adjusts his dancing to fit the tempo of music.
Shall we?
Now, he can dance without actually looking at her.
Does he like for everybody to join in? Shall we?
And he learned how to do this from watching the teenagers that raised him, who listened to the Backstreet Boys all the time. And they would dance like this. Okay? Okay. But you can see the rest of that online. All right? But the basic point I want to make is that many animals have good motor learning skills, like circus animals and so forth, but they can't learn how to imitate. And we found in the parrots, these areas involved in the motor control of learned sequencing behaviors surrounding those 7 song nuclei. And I believe, although no one's taken the brain of that animal, and I don't want to look at it, we have to actually take out the brains to look at these, in case you didn't realize. All right. But we don't have to do that for humans, you'll see in a minute. Here, but basically what I'm saying is that when they move their legs like this, we find that, or any form, that it's those brain areas around the song nuclei that are activated. What about dancing in the human brain? Well, a colleague of mine, Steve Brown, has used MRI imaging and had dancers put their feet on these boards here and dance out of sequence with their feet, like salsa or something like this, and asked what are the brain areas activated with dancing. And I'm just showing you here, I have to go through this quickly, but the brain areas involved in rhythmic dancing, called metric dancing right here, sit where I just showed you in that frontal section, if you remember earlier, I had a frontal section of the brain called Broca's area. It sits right next to Broca's area here, involved in speech control. That is, from this study and others, is showing that the areas involved in learned motor sequencing behavior in humans, like in the vocal learning birds, are also directly adjacent to the areas that control speech. Kind of makes sense, right? So when we started publishing this, I just thought I'd bring a little humor in here. Some articles come out, at least this was a better one. It wasn't about intelligent design. Actually, some of that stuff has anti-religious text stuff, that I showed you before. Now they're calling me a song and dance man. Although it's not me who's doing the singing, but I can do a little dancing. So I just told you I would come back to that part of my life, and it does make a difference when you have this artistic background about how you think about your work. So what about the connectivity? We know that in humans, I said there's this direct pathway from the face motor cortex, I'm showing you with this red arrow, to these motor neurons that control vocalization in both humans and song-learning birds that you don't find in chickens. In chimpanzees or macaques, you have an indirect pathway, not a direct one, whereas in other mammals you don't have any at all. But what about the pathways surrounding the motor pathways in a non-human primate? If you showing here in the bottom panels now are the individual cell types, color-coded, either white, red, or black. These are the cell bodies, the arrows here are the axons. That these regions here have, even though it's clustered in birds or layered in mammals, have quite similar cell types and similar connectivity and similar genes. I'll just give you an example here. The song nucleus called L-man, or man in songbirds, has one axon that goes into the basal ganglia, one axon that goes to these motor nuclei that control the production of the sounds. In the non-human primate or rodent brain, it has one axon that goes to the basal ganglia, and another axon that goes to these neurons that control the motor output for other behaviors. So what is going on here? I think that there is a deep homology here in the cortical regions, and that's led to what I call motor theory for vocal learning evolution. My argument is that brain areas that control vocal learning evolved out of a pre-existing system that controls movement and perhaps motor learning, not just movement, automatic and innate movements, but motor and learned movements. And that this pre-existing system is an ancient brain pathway that can be layered or clustered in its organization and is over 300 million years old. And what then I argue happens is that the vocal learning pathways emerge out of it using the design of that pathway as an ancient substrate. And that's how you can get similar structures in distantly related species. And I think this can be applied to complex traits in general, not just spoken language. But, you know, evolving a new pathway to dance better, or some other pathway that helps you fly better. And so I was showing you in more diagrammatic fashion here, instead of words. So I argued that all vertebrates have this innate vocal pathway involving a midbrain region and these brainstem areas that control the production of the innate sounds, with no forebrain control. And they have this motor learning pathway here, consisting of an anterior region and a posterior region involved in the control of motor learning behavior. And that there's a mutation in some axon guidance molecules that control neural connectivity multiple independent times to take, and what I argue is to duplicate this motor learning pathway in the brain. I think it's being duplicated during embryonic development in the uterus or in the egg multiple times to be hooked up to different motor neuron groups to control different muscles. Duplicate that pathway one more time and now hook it up to the motor neurons for the vocal organ, and you have a motor learning pathway for speech. And that's how I argue that humans and songbirds have emerged these vocal learning pathways. So that's an interesting idea. Well, I think it's interesting. And when I came up with this idea, I thought, wow, you know, this is really cool. You know, if it's really true. It's crazy if it is as well. But I thought I was the first one to propose something like that. You go back in the literature, and you're not. It turns out that there's a theory called the motor theory in speech, or gestural theory of speech. And that is, if you might have noticed as I've been talking for the last 45 or so minutes, yeah, exactly, I'm doing this. Who's noticed that? You know, I'm saying this. We gesture as we speak. We don't gesture as we listen. Right? When somebody asks me a question in another 5 minutes from now, I bet you'll gesture. You try to talk without moving your hands. You have to hold it in place. Okay? We don't gesture with every single sound that we make. And gesturing is learned. Right? Every language has its own gesturing. Italians have it much more than others. And children learn how to gesture before they learn how to speak. And gesturing actually helps child development. And it's been argued that gesturing and the brain pathways for gesturing are somehow linked to the brain pathways for speaking. And chimpanzees and macaques can learn how to gesture, can learn how to do sign language. So I argue for humans, it perhaps evolved out of the brain pathways involved in gesturing. And to put this in a more simple view, I argue that it's like gene evolution for those who've taken molecular biology or evolutionary courses. One mechanism of gene evolution is thought to take an ancient gene, shown here in the square, duplicate that gene in the chromosome, like you have 2 copies here. One copy, or one brain pathway in this case, as argued by 2 papers here, goes on to maintain its old function. This would be like the motor learning pathway, but now it's the old copy of the gene. The new copy of the gene will then have a new function and diverge from that pathway because it's been relaxed from selection because the old copy can maintain that old behavior. And so I think there's a general principle here of evolution of brain pathways and gene evolution. So last 5 minutes, I'm sorry, I'm going a little over time here, but we'll finish up. Genes involved. So I'm going to talk about, quickly go through 4 genes, some interesting findings we have. One is work that was done by a graduate student of mine, Rui Wang, who developed a computational algorithm to search for single, non-random amino acid patterns in genomes. With modern molecular biology techniques, the number of genomes being sequenced each month is increasing. And we had available to us 23 mammalian genomes that have been sequenced by NIH, NSF, and other groups. And he developed this algorithm to look for amino acids that are shared, and luckily all vocal learners were sequenced as well. So humans, dolphins, elephants, bats. Actually, dolphin wasn't that far along, so we looked at 3 of them, not the dolphin. And we looked for protein coding sequences that are shared amongst vocal learners, but not amongst the non-learners, or those that are amongst vocal learners only and totally different amongst vocal non-learners in all different kinds of combinations. And if you have protein coding sequences that are shared amongst vocal learners that are not in the non-learners, this would have to violate known phylogeny. It means that elephants and humans share more similarity than chimpanzees and humans. And did such sequence exist beyond chance? And the answer is yes. We came up with these 23 genomes. We looked at 3,000 genes. Out of those 3,000 genes, we found 16 that violate known phylogeny like this. And the top candidates were, in the brain, were axon guidance molecules that control neural connectivity. And one of them, my favorite here, is ROBO1. And all vocal learners that use the larynx to vocalize, humans, elephants, bats, have this alanine from threonine transition. And this threonine sequence is in a very conserved protein coding sequence here that's quite ancient, and you can find it back in platypus and chicken and so forth. What was a little disappointing at first is that zebra finches did not have that mutation, but we went ahead and looked at the expression of this gene called ROBO1 and its ligand called SLIT1 in these song learning nuclei, and especially for the ligand. What we found is that in all 3 vocal learning species, the mRNA expression of this gene, or its ligand, was downregulated in the song learning nuclei compared to the rest of the brain. All right? Now, again, they're separated by 300 million years from a common ancestor. A species more closely related to hummingbird, the dove, did not have such expression, nor did the quail, like a chicken. It did not have this whole downregulation here. What's interesting about the parrot has 2 subnuclei here. One of them projects to the rest of the other 4 brain song nuclei, does not show the downregulation. The other one makes that special connection that you find in the human brain that makes that unique connection out to the motor neurons that control the vocal learning, the learned vocalizations. And that is the one that shows the downregulation. And so what are we doing now? We're taking this gene and we're trying to manipulate it and trying to downregulate it in a dove brain or a quail brain. And we'll see what happens. All right? But it's not only that gene. We found another gene in our screening experiments. This gene is a gene called DSP1. It's also regulated by activities such as this EGR1 or zinc gene. But it's only regulated by activity in the song learning nuclei shown here. All right? Whereas in this auditory population here. But in the motor areas around the song nuclei, when the bird is hopping and so forth, you don't get regulation of this gene. And if you look at in hummingbirds and parrots, in their song learning nuclei, such as this one here or in the parrot brain, these 2 here, it's also regulated by singing behavior, but not elsewhere in the brain. So this gene has evolved in the last 65 million years to be specialized in the song learning nuclei. a gene involved in neuroprotection. And we looked at the promoter regions shown in these lines here. This represents DNA sequence. The promoter regions of these genes, shown by these hatch bars here, in hummingbirds, parrot, songbird, all have what we call, shown blown up here, repetitive microsatellite DNA. This is DNA sequence that has a repetitive structure to it, like GA, 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 for those who know the 4 nucleotides. And that's strange because most people call this junk DNA. There's a lot of repetitive DNA in the human genome. We call it junk. We're finding that the vocal learners have independently evolved repetitive DNA upstream of the protein coding region of this gene. At 3 independent times in the last 65 million years. And we think that repetitive DNA is somehow influencing the regulation of the gene to be special in the vocal learners. And that this is a way to select for a complex change in the gene regulation without changing the protein coding sequence. All right, another example of a common, what we call housekeeping molecule called albumin, a calcium buffering protein, upregulated in the song production nuclei. Here in songbirds, HBCRA. Here in the parrot, these two here in the hummingbird. The same nucleus. Also upregulated in the motor neurons themselves that receives the projection from the forebrain. You know, those motor neurons I talked about down here in the brainstem. And upregulated in those same motor neurons shown here in the human brain, in ambiguous. Also, that controls our speech, the white label there. We looked at non-human primate brains. You don't see this upregulation. You don't see it in that is in macaques. You don't see it in chickens. You don't see it in doves. But you see it in songbirds, parrots, hummingbirds, and humans. All right. Final example here. The FOXP2 gene. Some of you might have heard about this. A gene that's, when mutated in humans, causes a speech deficit called apraxia. We have found that in songbirds, in the basal ganglia region here called the striatum, the FOXP2 gene goes up when a juvenile bird is learning how to imitate a song from an adult male. And when he's finished imitating a song past puberty, which is basically 90 days old in the zebra finch, that gene goes down in the song learning nucleus. And a colleague of mine, Constance Sharp, they injected what they call a lentivirus that has the RNAi molecule that against this gene that will bind to the mRNA in the cell and prevent it from translating into a protein. So basically, knocking down the protein here, as you can see. All right, and here's a control RNAi that didn't knock down the protein. This is the protein molecule shown in the Western blot. And I'm going to now play some songs of a control RNAi. So here's the tutor, songbird, different from a canary. Oh, no. Here. Oh, darn it. Okay, in the answering question, I'll try to pull it up. This is not working. Basically, I'll try to imitate this. It's like something like this. And this is the son who now grows up as an adult who imitated like that. Okay. And here's another tutor, something like this. All right. Whereas with the knockdown of the FOXP2 gene in this animal, what happens? He goes something like this instead of or here. And here it's like something like this. That's how it's doing. All right, and we'll play it later on and see if I did it right. Basically, what happens? This is like an aphasia song. This is like what happens in human families that have this gene mutated. They can still speak, but they can't imitate well. And the same gene is having the same effect in songbirds. So I'm going to end off here with 3 slides and then open it up for questions. We have convergent genes and vocal learners. Vocal learners have convergent amino acid and differential genes involved in axon guidance and neuroprotection. Shown here is 5 examples of those genes that I just talked about. FOXP2, should be 1, global 1, and so on, axon guidance molecule. I think we need these genetic changes to form novel forebrain connections in the vocal learners. And why do we have convergent mutations for neuroprotection? I started to think about this. Songbirds like to sing a lot. Humans like to talk a lot. You go to the zoo and look at chimps or macaques and so forth, they're not vocalizing a lot. Once you evolve vocal learning, we vocalize a lot. Dolphins are singing all the time. And I think that what that means is that we're using these brain pathways a lot to give a lecture that goes about 5 or 10 minutes overtime. So basically, we love to talk. And because of that, neurons use up, 25% of your sugar per day, percentage, 25%. The brain is very energetic. And when those neurons are firing those electrical potentials and depolarizing and putting a lot of calcium into the brain all day long, you need a neuroprotection mechanism. And that's why I think neuroprotection has evolved. Is this a unique situation in songbirds or vocal learning animals? I argue not. As we have been coming up with these results, other laboratories have found convergence as well. Independent evolution of eyes 40 times, each time using the opsin gene and the PAX6 gene. Independent evolution of wing spots in insects. There have been 2 gains and 3 losses in Drosophila, and each time the yellow gene involved in that. Independent evolution of the electric organ in fish, twice in fish lineage, 2 different lineages, separated by 20 million years from the common ancestor, each time using a sodium channel gene that gets duplicated. One copy maintains the old function, the new copy then is used to create electrical pulses from electric organs. Independent loss of limbs in stickleback fish and mammals. Can anybody think of a mammal that's lost its limbs? That went back into the water? What? Whales, that's right, whales, they kind of have fins. But like sea lions and others, that's right. And so each time, what they found is that in these marine mammals that have gone back into the water, they have inactivated a transcription factor called PTX1 that fish also have inactivated. All right? So it's not just the peripheral organ that is converging, it's the genes that are converging. And finally, independent evolution of brain pathways for vocal learning, such as speech. So I don't think speech is so special in that regard. It's doing as all evolution does. So conclusions here on my last slide is that I think we have convergent behaviors and brain pathways for vocal learning that are associated with convergent changes in these genes. I propose that these pathways emerge by duplication and divergence from an ancient pathway involved in motor control and motor learning. That's a deep homology. It helps explain the motor theory of gestural origin of speech. And I propose that this duplication and divergence may be a general mechanism for a complex trait evolution. And this is my team here, and the way to do this, I argue, going back to culture and science, that we bring together a lot of scientific approaches, and we bring together a lot of people from different cultural backgrounds, such as African Americans, Native Americans, Europeans, and so forth. So to come together to merge all these ideas and art to come up with these hypotheses. And I will go ahead and end off there and open up for questions for those of you who Yes.
So, we have guinea pigs, and I don't know that they imitate pigs, but they certainly make beeping and squeaking all the time.
That's right, yeah.
So I wondered if, and I assume there's other animals that similarly make a lot of noise, and who did whether these neural protection factors show up in those?
Well, we haven't looked at guinea pigs per se, but let me paraphrase your question. There's some species that produce maybe only 4 or 5 different call types. And there's some out there that produce like 20, 30, or 40. All right? And I think quails can come up to 23 or so, something like that. But those sounds, those 20 to 30 sounds are actually innate still. And we still don't find in them a high level of these neural protection genes. But I still think it's possible for these 2 things to be independent, and that is, there could not be an animal that's an innate vocalizer that vocalizes a lot, but the neural protection genes will then become mutated and be overexpressed. All right? Yes? Somebody had a question over here?
I was a little curious about how you measure the connectivity and like the pathways when you have to take, like especially of the dancing or singing if you had to remove the brain.
Right. So the old-fashioned technique, old-fashioned when I was a graduate student, these things have progressed over time. Let's say from the time I was a graduate student up until about 2002. All right. The technique is inject a neural tracer in the brain of some guy. You can't do this in a human because then you got to remove the brain afterwards. All right. And then so you wait a week, 2 weeks, or so forth, and these molecules, lipophilic molecules that like to hug along the membrane, migrate in the membrane from the cell body to a location, somewhere else in the brain, and then you can visualize those colored dyes basically under the microscope. Now there's some new technique called transcranial magnetic stimulation, where you can use with MRI techniques, and you can stimulate a magnetic pulse on the person's skull right here, and you can see in an MRI that signal transmitted from one brain area to another. And if you give the pulse at a low enough intensity, you'll go along the axons that are connecting those two brain areas together. And that way you can actually start to tell the connectivity in a rudimentary fashion, at least in the human brain, without taking out the brain. Now there's some dyes that people were trying to inject in post-mortem brains, people who donate their organs to science. But these, in a non-living brain, basically, the dye doesn't transport any more than, let's say, 2 centimeters. And the human brain is bigger than 2 centimeters. Yes?
I know in the human auditory system, thalamic feedback is really important.
What kind of feedback?
Thalamic feedback.
Mm-hmm.
So is that seen in other vocal learners, and/or is it seen in vocal non-learners as well?
So the thalamic feedback, the auditory pathway, occurs in both vocal learners and non-learners. There's thalamic feedback also for the vocal learning pathways. So this area called DLM, you see here in the songbird brain, or analogous area here outside the song system, sends up, see this white arrow here, feedback back up to the forebrain. So that thalamic feedback is, I think, is part of a normal forebrain loop throughout the entire forebrain. But while you're on the auditory pathway, see, I talked about this projection coming out of the brain here that's unique in vocal learners in terms of its vocal connectivity. There's a whole other group of scientists who argue that the connection between the auditory areas and the anterior Broca's speech area is what's really special about what you need to have spoken language. The problem with that argument there is that there's no evidence that other species don't have it. And it's recently, with transcranial magnetic stimulation actually, it's recently been discovered that chimpanzees have this connection as well. It's just smaller relative to the rest of the brain than it is in humans. In humans it's more developed. Whereas chimps and macaques don't have at all the connection from the forebrain to the motor neurons. So I'm of the school of thought that it's really forebrain to brainstem connection and not the thalamic input or the auditory motor connection that's special. So another hand over here.
Yeah. I was just wondering, since you said the learning with the motor skills is similar to the learning, vocal learning, is there a similar time frame with learning languages as with learning motor skills? Like, can you learn motor skill much better and completely if you learn it in the same pattern as the language?
The answer is so language is not the only thing that has critical periods. There are a lot of behaviors that have critical periods, and they don't all have the same timing to them. And what I've noticed, and it's not just me, is that if you look throughout the literature, these studies of when these critical periods occur for learning how to speak, learning how to read, learning how to walk, learning how to dance, that kind of thing, learning how to play the piano. All these critical periods, they seem to me is to coincide with the phylogenetic appearance in the human lineage or in the animal lineage. That is, it's easier to learn how to walk, which is a more ancient behavior, than it is to learn how to speak. It's easier to learn how to speak, it comes naturally to us, than learning how to read, which is thought to have something to occur sometime in the last 5,000 years. I think that has something to do with changes in how new the connectivity is. The system is still evolving before it really sets in to have, you know, more automatic learning, so to speak. Does that answer your question? Yeah. And even though you didn't answer this question, I have an answer for it, which I'm going to answer it for you, ask you. Is that, what is the difference between those brain areas where in the songbirds we found that there's this critical period for learning how to sing is different from the critical period to learning how to, let's say, do some other motor learning skills with the wings, where learning how to fly is easier. What we're finding is not the neural connectivity that differs, but it's certain genes that are expressed that allow synapses, the neurons, to communicate with each other more stronger or more weaker. What happens in the other brain areas, learning how to fly or learning how to walk, those connections set in quicker. And the genes that are stabilizing those connections are shut down sooner. Okay? Whereas, or shut on sooner, put it that way. Whereas in the vocal learning areas, there's more of a delay in those genes. So in order to prove that hypothesis, what we're doing now is trying to take those genes and either downregulate them or upregulate them to see if we can reopen the critical period for vocal learning in these animals. And if we can do that, then we can temporarily reopen the critical period for learning lots of things. But you don't want it open entirely because babies always forget. And that's why you need to close it down at some point because you'll forget everything you learn. Right.
So you can make patients that can sing?
Trying. Yeah, well, I don't know how far we'll get, but if I can get halfway there, yes, that's right. Yeah.
When I studied evolution, we reviewed the different hominid species from Homo habilis to Homo erectus to Homo sapiens, and the progressive increase in the size of the cerebrum, which we were told was correlated with increased capacity for abstract symbolic thought and so forth. Is that model also rejected? Has it been discarded?
I would
you
say.
seem to be arguing it's really a matter of connectivity, not a science.
Right. Actually, I learned as a scientist it's best not to have absolutes because there's always some exception to the rule. And so I would argue that size is important up to a certain point. And it is not as important as in terms of magnitude as we'd like to think about it, in terms of complex behaviors. So I would say, if I were to put a percentage to it, maybe size matters about 15 to 20%. The other 80% is basically connectivity in the individual genes. Right? So why do I say that? Because animals, let's say birds that have very good visual systems, like a hawk and so forth, can see a squirrel from way up in the sky and dive down and eat it, has a bigger visual pathway or forebrain areas than the ones who aren't good at vision. But the relative increase in that size is not as important as the change in neural connectivity that allows more rapid neural firing between these brain areas. So I would say that for that reason, the size of our brains are overrated. And when I see some of these lectures, I've had debates with anthropologists who show these human models, what is it, Neanderthals and then Australopithecus and so forth, to show this, sort of Scala-Natra progressive trend to the, humans and who knows, they went for Europeans and so forth, like the others are doing. That's a joke. So anyway, but basically these models where they talk about brain size leading up to the human lineage. But if you look at the more ancient hominids, they're smaller body than us. And they have to have smaller brains because they have smaller bodies. And a lot of these anthropologists do not take that into account and do not realize
that. Trajectory was not a simple brain size, but increasing proportion of the cerebrum.
That's right. That's right. Yeah. I haven't seen that in the hominid evidence yet for increasing portion of the cerebrum. In relative proportion to the rest of the brain. Yes?
I do have a question, but just to that point, Gary Lynch's book, Big Brain, I don't know the Latin name, but he talks about a hominid that had a bigger brain than us that actually is extinct. So.
Well, well, actually, Neanderthal brains were bigger than humans.
So that kind of goes against
Yeah, right, exactly. And the anthropologist who has subscribed to this view, when they present the Neanderthal data, they normalize the body size. When they present the Australopithecus data, they don't. All right? It's interesting. I mean, I won't name names, but some very famous people. I debated with them on this, and they said, well, they're not enough of the skeleton to normalize appropriately to the Australopithecus. And if you do it in the Neanderthals, you got to do it with the Australopithecus.
It's the same one.
Yeah, exactly.
I just want to ask, do you see, is there a relation between the size of song nuclei in different species like hummingbirds and
Yes.
parrots?
Yes, yeah. Okay.
Some complexity.
Right. Yeah, so that gets back to the 20%. So this green, purple, blue labeled area, it is true that in most vertebrates, that part of the brain evolves last, not just in humans. And the bigger it is, relative to the rest of the brain, the more complex behaviors it tends to have. Well, it turns out in the vocal learning birds, the auditory pathway, if you remember, was sitting in the back of the brain here, a blue labeled region on the other side. That auditory pathway is bigger in the species that can imitate than the ones who can't. All right? Not much, but it is a little bigger. And then you asked about the song learning nuclei. Well, this purple labeled region here, the area X, in species that are more stereotyped, relative, that area X structure relative to the one above it called man, the cortical region. The cortical region is bigger relative to the strident in species who are producing more complex songs, including the parrots that can imitate human speech. Whereas in the species that are producing more stereotype songs, the purple region, the striatum, is bigger. And what is known is that when you lesion the striatum, animals or humans, we can't actually crystallize that well onto a song. We can't actually, we always forget, basically. Whereas if you lesion the cortical region, we're always stereotyped. So what's thought to happen is that these two regions, one is putting plasticity, the other is putting stereotyping into the behavior, and the two come together in order to enable vocal learning. Because if you have complete plasticity, you can't learn. If you have complete stereopticity, you can't learn. And so I think those species that are more stereotyped or more verbose imitators have taken advantage of that to have more complex behaviors.
Take one more, Fred?
What about birds like cowbirds that lay their eggs in nests in other species?
Yes.
Where do these kids learn from? Do they speak different languages?
Right.
They
So.
learn later in life.
So, yeah, that's interesting. So like I said, nothing's 100%, but for those who don't know what he's saying, I gotta explain it. Cowbirds are actually songbirds, but there are other birds that are non-songbirds that do this as well. They lay their egg, the female will lay an egg in another species' nest, and when that egg hatches, that little nestling will imitate or produce the calls of the other species, and produce it louder and more often, such that the mother will feed the other species instead of its own young, and its young will sometimes die.
And then the cowbirds find each other to mate if they're speaking, if they
Right.
talk to each other.
Right, so this is the other 20% of the equation. So, yeah, how is that cowbird going to find its mate once it fledged nest, or find its own flock? And all vocal learning species, including humans, have an innate predisposition to be attracted to their own species vocalizations. Yeah. So we are born with the ability to produce about 400 phonemes as an infant, as a child, soon after birth. Those are the innate sounds. We learn to sequence them. But those sounds have certain they have certain sounds to them. They're human sounds. And so, for instance, a mockingbird. Mockingbirds when I say we, I mean the scientists that study mockingbirds, count that an individual animal singing anywhere from 1,000 to 3,000 songs.
Including cell phone rings.
Right, that's right, the mechanical sounds. But how does another mockingbird recognize it's another mockingbird if it's imitating cell phones, right? And what mockingbirds do innately is that they sing their songs 2 or 3 times in a row, like. And then they'll do. And as long as they do it 2 to 3 times in a row, the others will recognize, okay, that's a mockingbird. But the individual sounds will be imitated. And this is something similar for cowbirds.
So on those notes.
I told you it was a long
You
explanation.
have even more skills than we
All
realize.
right, okay.